Right. Okay, so I was asking whether that fruit fly you made with the red eye would have been literally the first red-eyed fruit fly.
Well, no. What I should have said down there, so the piece of DNA that we're putting into the fly has that transgene, that new gene, which has the green fluorescent protein fused to whatever protein of interest it is that you're studying. On top of that, there's a second piece of DNA, and that piece of DNA encodes for a protein that gives the flies red eyes.
Okay.
Because that way, all you have to do is screen through the flies that you've injected, looking for the ones with red eyes, and then you know that the ones with red eyes will also have the fluorescent gene.
I see.
So the first fly that I ever made, which was called Kevin,
Yes.
so Kevin carried...
this might be Kevin's song. Yeah.
Kevin carried a gene which then encodes a protein called... I don't remember back now. It doesn't matter, because it was a protein called CP60, but CP60 means nothing to anybody.
No.
Doesn't really matter. Anyway, so maybe it's that. Maybe I'll put it abnormal spindle. So this is protein which is in fruit flies, but it's also in humans.
Oh.
And if you have mutations in it, in humans, it causes a small brain, causes microcephaly.
Yeah.
And it does that because it's important in cell division, in development, most specifically in the brain. Okay. So I took the gene for abnormal spindle, ASP, intrasophla, and I fused it to that GFP. And I made this piece of DNA that had that, plus it had this little red eye gene.
Yeah.
That's the one that I injected into a batch of embryos.
Yeah.
So like probably 600 embryos, and of those...
One by one?
Literally? Yes, yeah.
Oh my
God. You kind of do 30, kind of do 30 an hour. So it kind of took me 2 days to do those injections. Yeah. And then 2 weeks later you have to screen them.
Yeah.
When they've grown up into the adults, and of those 600, one had red eyes. And that
you
was
must
Kevin.
have been so relieved that Kevin was there.
So then obviously Kevin was a boy, so he's going to die eventually. So I had to then cross Kevin to one of our standard flies, females, so that all
Did
of
you
Kevin's...
literally put two of them in a romantic
Well, actually,
situation?
it was 3 females plus Kevin.
Oh, right, I was
to
going to
give
say.
him the best chance. Right? So then Kevin's offspring then all have that, well, actually half of Kevin's offspring, because you've got chromosome pairs, and we put this piece of DNA just on one chromosome.
Yeah.
So half of Kevin's children have the red eyes and this green fluorescent protein linked to ASP, and half of them don't.
Right.
So we have to take Kevin's children, the orange-eyed ones that have this gene, and we have to cross them to each other. We do sibling crosses.
Oh dear. Yeah.
I know. Well, they're not humans, are they? And then
I'm sure it happens a lot in the wild as well.
And so then their offspring now have 2 copies.
Yeah.
So that's stable.
Yeah.
So it's Kevin's...
that's what you mean by stable.
Yes,
okay.
So Kevin's grandchildren are the ones that we then image.
Yeah.
And they're the ones that we would put on the shelf ad infinitum. So Kevin's offspring. So I still have Kevin, but not Kevin.
Yeah.
I have and however many generations.
Oh,
so that was
gosh.
the first fly that I made. So that was in a PhD. So that was 1996.
Oh, wow.
Was my first fly. So that means that Kevin's offspring have been in the lab for however many years.
27 years.
27 years. So at 1 generation per 2 weeks, right, which is what, 25? Call it
25 times
generations a year.
27.
25 times 27. 500, nearly 6. No, 6.
No, double that, isn't it? Oh, I don't know.
3 times 3, 30. Hang on. So what were we doing now? 25 per year.
Times 27.
Yeah, times 27.
No, I think you're right,
yeah,
actually.
it's about 5. Yeah, probably 5 to 600
Generations later.
600 generations
Wow.
down the line.
Wow.
And Kevin's still there in my mind.
There's so much in this, so much material. I'm going to be spoilt for material.
So when we take our embryos, ready to image them.
I'm just going to start a new